Okay, Assalamu alaikum everyone. My name is Rasul Turko. I'm a third year medical student. And today we're going to be talking about the fatty acid oxidation and ketone body synthesis. So, inshallah, it'll be an easy lecture. I tried to compress all the high yield information and everything they usually ask in the exam into my slides. Hopefully, they're going to be more than enough for your exam. As I said, inshallah, it'll be an easy lecture. I love this lecture. I love this lecture. It's pretty straightforward, and I think Dr. Abdul does a great job of really like talking about the high yield stuff that will most likely come. So for objectives, it's gonna be the same objectives. We're gonna be talking about again uh, fatty acid oxidation and ketone synthesis, and we're gonna also mention a couple of different uh, diseases that happen or dysfunctions that happen due to the oxidation. I usually include some questions at the end of my lectures, but since this is a recorded session, I didn't see the point of putting it since like no one's going to be answering my questions. So therefore, like I would recommend going through Lippincott or integrative biochemistry to really test your knowledge when it comes to this lecture. So okay, Bismillah. So first of all, we're going to be talking about the lipoprotein pathway. So when we talk about fatty acids and how they're oxidized, one thing to know, one thing to really understand is um, how fatty acids are like ends up in our body. So there is two pathways: is either endogenous, where our body produces fatty acids, or exogenous, when we eat uh, different meals that have fatty acids. So in this pathway, we're going to be talking about exogenous mostly, since we're going to be talking about how we break it down and how we utilize it for energy later. So first of all, this is not really as important as you guys may think in this lecture specifically, because later in Mol 2, there's 2 to 3 lectures that are going to be talking about the insulin glucagon, and there's multiple different lectures also that are going to be talking about the whole pathway of fatty acids, how they get broken down, like, and then you guys are going to get into the tiny details of that. So this is just an overview. It's not as important again for this lecture, but I'm still going to go through it like briefly. So first of all, here, doctor talks about how we talk about dietary fat. So this is when we eat fatty meal. What happens is, first of all, everything that's digested in our small intestine gets absorbed as a chylomicron. So chylomicron is the one is the entity that relates to the endogenous kind of fat that we eat. So this chylomicron goes through the capillaries, and in the capillaries we break it down into what's it called, VLDL. Sorry, yeah. So the chylomicron here gets broken down in the capillaries, and then what a chylomicron consists of is a triglyceride, like a triglyceride, a cholesterol in the middle, like right here in the orange in the middle, and then a blue border, which is the hydrophilic layer. So what we care about here is the triglyceride in the middle. What's a triglyceride? Again, triglyceride is going to be TG or TAG, triacylglyceride. So they're the same thing, and it's important to know that these two are the same thing. We use them. Doctor uses them interchangeably, and most of the textbooks use them interchangeably. So let's say we have the chylomicron, we have the triglyceride in the middle. It gets broken down in the capillaries or in adipocytes. But here we're going to be talking about the capillaries, and in the capillaries it gets broken down to glycerol, fatty acids. The glycerol then ends up being used in gluconeogenesis in the liver, and the 3 fatty acids get broken down into energy, and that is exactly what we're going to be talking about here. So the whole pathway of what happens to the chylomicron after that, doctor like briefly talks about it, but again, for this lecture is not as important. So it just basically says that after it gets broken down, goes to the liver. In the liver, it gets changed to VLDL, and the VLDL, if you notice here, the white stuff is not as much as in the chylomicron. That means we have less fatty percentage in the VLDL than we had in the chylomicron. Again, gets broken down in the capillaries, becomes an IDL, which is intermediate density lipoprotein, goes back to the liver, and then becomes an LDL. So that whole thing, that whole pathway, we're going to be talking about. We're going to be talking about later in later lectures. For now, what we need to know is when we eat a fatty meal, we get a chylomicron. A chylomicron has different components, has triglyceride, has cholesterol, and has a hydrophilic layer, which has proteins and phospholipids around it. And that chylomicron gets broken down in the capillaries or in the adipocytes. Gets broken down in different parts of the body to give us 3 fatty acids and a glycerol. Okay. So as we said, glycerol is used in gluconeogenesis, and the fatty acids, when they get broken down, acetyl-CoA, and that is what we need. That is what we need to use in the citric acid cycle to give us the ATP. Remember, like in Mol 1, we talked about the whole citric acid cycle, which has like different amino acids, and then that whole process gives us, if I remember, NADH, FADH, CO2, and ATP. So the whole point of fatty acid oxidation is we get acetyl-CoA. The acetyl-CoA gets utilized in the citric acid cycle, and then we end up getting ATP. And a very, very important thing to know or notice here, and that's the way I like to think about it when it comes to fatty acids and glucose and all these things, is when do we need to break down fatty acids? So we need to break down fatty acids when we need energy. Again, because fatty acids gives us energy. When do we need energy? When we have low glucose, low blood glucose, when we don't have as much glucose in our body. So that means we break it down when there is a hypoglycemic state, because again, when our body needs energy, its primary what it goes to do primarily is to break down the glucose in our blood. When we don't have glucose in our blood, that's when it goes and attacks, or that's when it goes and utilizes the fatty acids. So a very, very important note is fatty acids synthesis happens when there's hypoglycemia, and that happens when you can imagine when there's fasting, for example, or when we have a prolonged exercise period, when we're exercising a lot and we haven't eaten, that means we don't have blood glucose, or we don't have glucose in our blood, and that's when we go and break down fatty acids. And that's a very, very important thing to distinguish. Then it helps, because that helps you understand when and what enzymes helps us in that process. Okay, and we're going to talk about that later. Okay, so as we said, triglyceride, TG or TAG is the same thing. That is our primary focus, because triglyceride has 3 fatty acids and a glycerol. So here the author talks about triglyceride in the capillaries, and then here he goes and talks about how it gets broken down intracellularly. So in the beginning, we talk about how it gets broken down in the capillaries. So this image here is literally right what we talk about right here. Okay, so first of all, when we have a triglyceride, and it goes through our capillaries, we have things coming out of the endothelial cells called the heparan sulfate. These little like gray things. These are the heparan sulfate. These heparan sulfate come out of the endothelial cells. They are attached to something called LPL. LPL is very, very important. It's a lipoprotein lipase, and from the name lipase, ase means it breaks down lipids. So it breaks down lipids in the triglyceride. So LPL is connected to the heparan sulfate on the endothelial cell. This LPL targets the triglyceride. But there's something that the triglyceride has that the LPL kind of attracts, and that is a apoprotein C2. So LPL goes and goes and like finds the apoprotein C2 on the triglyceride, and that helps it break down the triglyceride into different things, again, fatty acids and glycerol. Okay, so again, this is a side note to the lecture. This will be talked a lot about talked about a lot later. For now, all we need to know is that LPL, which is on the heparin sulfate, attaches to the triglyceride, to the apoprotein from the triglyceride to break it down. Okay. And here is when we start talking about actual beta oxidation or fatty acids. It's the same thing again. Fatty acids is the same thing as a beta oxidation. It comes from the name. Beta oxidation comes from the carbon beta. There's a beta component of the carbon, so it comes from that. So here's the first step. So again, when we are in a state of hypoglycemia, our body has stored fat, right? Already. So that fat is what gets utilized during those levels of hypoglycemia. So we imagine it like this: when we have adipocytes, again, those fat cells have all those fatty acids, and when our body senses that we don't have enough ATP, we try to go and break down glucose or glucose storage that we have. If we don't have glucose storage, since we haven't eaten in a while, that is when we go through. That's when we go to fatty acids in the adipocytes to help us get the oxygen. Get the ATP that we need for our energy during the day. So here we talk about an fatty acid oxidation in the adipocyte, in the cell itself, intracellularly. So first of all, here I'm going to start with the whole process. Then we're going to kind of read what's going on the left. So first of all, when we're in a stage of hypoglycemia, would we have a high level of insulin or a high level of glucagon? Obviously, it would be glucagon because insulin is produced when we have high levels of glucose. So insulin comes out and kind of tries to like store that glucose that we have. So when there isn't a lot of glucose, we don't need insulin. That means the glucagon, which is kind of like the opposite of insulin, that's when it gets used. So glucagon actually activates a receptor, a G protein receptor, and then that activates GS. Again, this is not important in this lecture. What's important is that glucagon kind of activates a G protein receptor in the cell. That actually produces cyclic AMP and pKa. We've already learned that from mole 1 that ATP produces cyclic AMP. So here what we need to know is the glucagon produces pKa, which is protein kinase A, and cyclic AMP. So we have protein kinase A and cyclic AMP now. What do you think protein kinase A does? So I've learned this also during mole. It took me a while to understand that happens, but I think during mole 2, I've realized at that point that a kinase phosphorylates something always. So a kinase phosphorylates something, and this is really easy to deal with when we have anything like this kind of reaction here. So when we have protein kinase A here, we are expecting that it's going to phosphorylate something. So pKa here phosphorylates two things. It phosphorylates something called perilipin, and it's those little like purple things here, and it phosphorylates something called HSL. So LPL was a lipase that broke down triglycerides extracellularly in the capillaries, and intracellularly we have something called hormone sensitive lipase that does almost the exact same thing, which break down the triglyceride. Okay, so phosphorates two things. It phosphorates HSL, and then it phosphorates something called perilipin. So when it phosphorates those two things, it activates them basically. Okay, so HSL we're going to talk about in a second. Perilipin gets activated. When perilipin gets activated, it's connected to this thing called CGI58. This green thing. So when perilipin is activated, it lets go of that CGI. Okay, CGI goes when it leaves the perilipin complex. It activates ATGL, and ATGL stands for adipocyte. Where is it? ATGL. Adipocyte triglyceride lipase. Where I'm just trying to find it right here. ATGL, yeah, ATGL, adipose triglyceride lipase. Okay, so it activates ATGL. And what does ATGL do? ATGL is the primary enzyme that starts breaking down the triglyceride. So it breaks down triglyceride and makes it a diglyceride or a diacylglycerol. So it's the same. So again, a triacylglycerol and a triglyceride is the same. Okay, so it takes it from triacylglycerol to a diacylglycerol. Okay, so it's the first enzymes that really works. After that, we now have a diacylglycerol. We get the hormone sensitive lipase that we've already activated. And that hormone sensitive lipase breaks down diacylglycerol to a monoacylglycerol. Okay, and then after that we have a monoacylglycerol. We get an enzyme called MGL, which is a monoacylglycerol lipase, and breaks down to monoacylglycerol to what we need exactly: the 3 fatty acids and the glycerol. Okay, these 3 fatty acids then go through the capillary and get transformed by a protein called albumin. It gets transported by this protein to the liver, and then the glycerol also gets transferred to the liver and then gets used for gluconeogenesis in the liver. Okay. So I'm gonna try to read this and make sense of it. So when we have again glucagon and epinephrine, those 2 things activate hormone sensitive lipase. This also kind of makes sense in my head. This is how I think of it. I've made kind of a connection between glucagon and epinephrine, since epinephrine is kind of produced when there is a fight or flight kind of situation. So this is in my head. This is how I think about it in my head, is when someone has had we're having a flight or flight experience, or they might be like going through a prolonged kind of exercise, so they have a lot of epinephrine. That is when it gets activated and we get hormone sensitive lipase. Does that make sense? Glucagon is the same thing. Since it's not insulin, since we haven't eaten recently and we don't have high number of glucose in our blood, insulin is not produced. And since insulin is not produced, glucagon is going to be the one that's produced, and is going to be the one that is activating the hormone sensitive lipase. Okay, so again, it produces cyclic AMP and PKA. PKA activates perilipin and HSL by phosphorylation, so it phosphorylates those 2. CGI dissociates. This itself activates ATGL. From ATGL, we get a diacylglycerol. Then we have HSL, which gives us monoacylglycerol, and then those fatty acids get transported by albumin to the liver, and then glycerol is also transferred to the liver, where it's used for gluconeogenesis. Okay. And here is important to know that insulin here does the opposite as glucagon, so it would not activate a phosphorylase like PKA, for example. It activates a phosphatase. So it activates a phosphatase, which would break down, which would basically which would take the phosphate out of the HSL here. So a phosphatase, as I said, it takes the phosphate out. A phosphorylase adds phosphate. Phosphatase takes out the phosphate. So when we're taking out the phosphate, it inactivates it. It activates those enzymes, the perilipin and the HSL. Okay, so obviously insulin would do the opposite of glucagon, and it would make sense because, again, when do we get a high number of insulin? When we have we have recently eaten, and then we have high number of glucose in our body. That glucose is the one that we need for energy. So at that point, we would not need, when there's a high number of insulin, when there's high glucose, that means we have insulin. At that point, we would not need to break down fat because we have glucose for our energy. The only time we need, or the only situation where we would need fatty acid synthesis, is when we don't have enough glucose for our energy storage. Okay, so here insulin actually promotes fat synthesis, not fat. not fat oxidation. Okay. So this is the first. This is the very first step. Is basically breaking down that big triglycerol uh, complex into fatty acids and glycerol. Okay, and this all happens again intracellularly. So it's all happening inside of a adipocyte, and that's important because basically everything, this whole fatty acid oxidation happens in the mitochondria. So it's important to note that the fact that this is all happening intracellularly. Okay. So here we talk about the glycerol. Again, what we said the glycerol does is the glycerol goes and gets activated or gets used up, utilized when we're trying to make glucose, gluconeogenesis. So I've mentioned this to show you the fact that fats, which is the glycerol, aka glycerol in this situation, uh, is a source of carbon. So the carbon, the carbons we have in this fat molecule is the one that is produced or that is utilized to produce glucose or in gluconeogenesis later on. So glycerol would have carbon uh, atoms or carbons that would get utilized in gluconeogenesis. I remember we had a question about this in one of our exams. I don't know if it was the midterm or final. This is why I felt the need to mention it in my lecture as well. It's because I thought it would be high yield. Um, for these kind of enzymes, I don't remember it being high yield for our lecture. So since this whole like since you guys know, or as long as you guys know that the glycerol gets used later on for gluconeogenesis and to make glucose, I think that's enough. We don't need to know the process of how glycerol does that. And I haven't seen during my 3 years at Al Faisal, I haven't seen a lot of questions about, if any, about glycerol and the different enzymes. Okay, so now so now that we have a fatty acid or three fatty acids, now the process of breaking those fatty acids starts. So first of all, a fatty acid needs to get activated. So it gets activated by something called acylation. So what it means is we literally just add an acyl CoA to the fatty acid. So instead of it being a fatty acid, it becomes a fatty acid CoA. That's literally what they mean by activation, and we need that. We need that for it to be for it to, we need that to ease the process of entering the mitochondria. So there's different processes that we need to do in order for the fatty acids to enter the mitochondria and get broken down. This is one of them. Is it getting activated by an acylation of CoA. Um, Again, this reaction is not really important. I've added it to just reiterate the fact that CoA gets added to the fatty acid right here. So it becomes a fatty acid, becomes a fatty, sorry, right here, becomes a fatty acid CoA, basically. Okay. So that's all that happens. This is it. This is how it gets activated. One enzyme to note over here also that's very important is a long chain fatty acyl CoA. Oh, it's right here. Yeah, perfect. Fatty acyl CoA. This is what it becomes. So a long chain fatty acyl CoA synthetase, and what it does is it combines the fatty acid with an acyl group to help with oxidation. So that whole process is catalyzed by the enzyme LCAS, long chain fatty acyl CoA synthetase. And then here, doctor also mentioned one of the diseases that is due to a deficiency in LCAS, and it's adrenoleukodystrophy. So that is basically when that is basically a deficiency in LCAS, so a long chain fatty acyl CoA synthetase. So when we have a deficiency in that, that means we are not able to add the acyl CoA to those chains of fatty acids. So when we don't add the acyl CoA, we're not activating that fatty acid. So we end up having an accumulation of fatty acids or a long chain fatty acids. This buildup of long chain fatty acids in the brain adrenal cortex ends up giving us adrenal leukodystrophy. Okay. It hasn't really talked about it. Other than that, he hasn't mentioned symptoms and different and what it presents like. He only mentioned the fact that there is this disease and it's due to a deficiency in this enzyme. That's all he said, and that's all we need to really focus on. We don't really need to get into the specifics of this disease. Okay. All right. Step 2. So at this point, we've said again, we had triglyceride. We broke it down by pKa, by different enzymes, but pKa was the main factor here. Actually, let's do a review so far. So we had a triglyceride. That triglyceride gets broken down by triglyceride gets broken down by hormone sensitive lipase, ATGL, MGL, into 3 fatty acids and glycerol. This fatty acid then gets activated by LCAS, and basically gets an acyl CoA. So it becomes a fatty acyl CoA. And now we are back to step 2. So at this point, we have a fatty acyl CoA again, and it's in the cytosol of the mitochondria. So we needed to get inside the matrix. So we needed to get inside the inner. We need to get in the mitochondrial matrix. So in order to do that, we have different enzymes that it goes through. So now we have fatty acyl CoA. We need to add a carnitine on it. So adding a carnitine on it by an enzyme called CPT1. Now we have an acyl CoA carnitine, or acyl carnitine. This acyl carnitine can easily go through this layer and get inside the mitochondrial matrix. So it needs a carnitine. So first of all, it gets activated and then gets a carnitine, and that carnitine eases the transport of it inside the mitochondrial matrix. So CPT1 adds a carnitine on it. Now here we have a translocase. A translocase just takes the acylcarnitine from the mitochondrial from the cytosol to the mitochondrial matrix. So it brings it all the way inside the matrix. And now that we have the acylcarnitine, we need it. We don't want it as an acylcarnitine. We want it as a fatty acid. So now that's when CPT comes and removes the carnitine from that complex and makes it a fatty acyl CoA again. Okay, so it's kind of so we have inside the matrix what we had outside. Does that make sense? It's just we needed the carnitine to make it to allow it to get in. Okay, so again, I'm gonna go over again. So CPT1 adds a carnitine to it. Carnitine gets through the border by an enzyme called translocase. Now that we have an acylcarnitine inside the matrix, we use CPT2 to remove that carnitine out and end up with a fatty acyl CoA by itself. So an important thing to know is, so we'll do a little recall from the last lecture. We were fatty acid synthesis, where we talked about how in order to make fatty acids, we need something called a malonyl CoA. So this malonyl CoA is kind of the precursor of fatty acids, and knowing that or recalling that will make it easier to understand why a malonyl CoA inhibits CPT1. So obviously, when we have a high number of malonyl CoA, when we're making fatty acids, we would not need to be breaking down fatty acids as well. So when there's a high number of malonyl CoA, that inhibits CPT1, and inhibiting CPT1, it kind of pauses the process of breaking down fatty acids or beta oxidation. Okay, and this is also high yield. It's as we talk about it in different diseases later on. But for now, what we need to know is malonyl CoA inhibits CPT1. making it harder or making kind of like stopping the process of rate oxidation. And then we move on. So step 3 is the actual rate oxidation of fatty acids. So here, doctor kind of just like put all the enzymes and put like different kind of characteristics of every enzyme. I don't remember this being high yield as the stuff that we went on earlier. But let's go through it. So first of all, there's the oxidation process. So there's 4 reactions: oxidation, hydration, oxidation again, and then cleavage. So first of all, goes through something called oxidation, and that is when we basically just add an oxygen to what we have. That is due to an acyl-CoA dehydrogenase. Okay. Family we'll talk about in a second, but it's an acyl-CoA dehydrogenase. This is kind of the like most common enzyme out of those 4. Because yeah, the other 3 aren't high yield as much as this one. But yeah, so there's an acyl-CoA dehydrogenase as in a family. I'll talk about it later. Then we have a hydration that happens through anyl-CoA hydratase. Again, hydratase. Third reaction is 3-hydroxyacyl-CoA dehydrogenase. So again, dehydrogenase, those 2 are dehydrogenases, and these 2 are oxidation. So one is an acyl-CoA dehydrogenase, and one is a 3-hydroxyacyl dehydrogenase. Fourth enzyme is a B-ketoacyl-CoA thiolase. Okay, those 4 enzymes. I've only mentioned stuff that might be important to you. Again, it's not as important as the other things, but I only mentioned it here to kind of just in case, like, because the doctor also mentioned it, so I thought I'd mention it too. So in the oxidation, we reduce FAD to FADH2, and in the second oxidation, we reduce NAT to NADH. Just keep this in the back of your head in case they ask you about what an oxidation reaction does or what this dehydrogenase reaction does. Even though they haven't done that. They haven't done that in my batch, and I haven't seen them do that, just in case though. And then when we talk about family. So what do we mean when there's a family of enzymes? So again, when we have a fatty acid is not just like one acid by itself. It's a long chain fatty acid. So by what we mean by that is we're talking about like 12, we're talking about 14, we're talking about 16 fatty acid, like a 15 chain long fatty acid. And how does that get broken down? It doesn't just go through this from 16, it becomes 1, and then we're good. No. So what happens is it goes through, first of all, it does go through the breakdown, as we said, and then it does go through the membrane, and then gets into the matrix. After that, now we have a 16 chain long fatty acid. It goes through those 4 different reactions, but then every single time it goes through this reaction, it loses 2 carbons. So yeah, I wrote it right here too. So every round we lose 2 carbons. So first of all, we have 14, goes through those different 4 reactions, becomes 12. After that, it goes through the reactions again, becomes 10. You guys get the point. Until we have no carbons, and that's when we actually have broken down that whole long chain fatty acid. We can use it for ACP later. So it's important to know that this reaction keeps happening until we keeps happening, and every time it happens, we lose 2 carbons. And then I mentioned that talk about the family. Yeah. So when we get a 14 carbon fatty acid versus a 12 carbon fatty acid, we're not going to use the same enzyme, right? We use every single long chain fatty acid needs a different kind of enzyme. So a 12 carbon fatty acid needs an enzyme here. This one would be called a 12-acyl-CoA dehydrogenase. Okay. When we have a 14 carbon fatty acid, we would need a 14 acyl-CoA dehydrogenase. So that's what we mean when we talk about a family of enzymes. So it has different enzymes, each for a different number of fatty acids, and then that is when we use it. Does that make sense? Okay. So that is literally the beta oxidation. So it's literally 3 steps. It gets, first of all, broken down to fatty acid glycerol. Fatty acids then get transported inside the matrix. From inside the matrix, we have different enzymes, 4 different enzymes that break it down. Every cycle it goes through, we break down, we take 2 carbons out until we get 0, and that's when we have completely broken down fatty acid into carbons, and then those carbons can get used in the ATP for ATP later on. Okay. So here, I mentioned CPT1, clinical significance. So what did we talk about in CPT? So CPT happens, what does CPT do? So it's an enzyme that adds a carnitine onto the fatty acyl-CoA to ease the transport inside the matrix. So again, when would CPT be active? CPT would be active when we have hypoglycemia, or when we have low glucose in our blood. That is when we need fatty acid oxidation, and that is when CPT starts working. So when we have something like hyperglycemia in our blood, would that mean that we're using CPT1, or CPT1 is active during that? No, because we wouldn't need fatty acid oxidation when we have high blood glucose in our blood. When we have a lot of glucose, when we have hyperglycemia, we already have enough glucose to break down for ATP. That means we don't need fatty acid oxidation as well. So CPT will not be used. So why is that significant? Because CPT1 dysfunction happens in type 2 diabetes and insulin resistance. So what do you mean when we have type 2 diabetes? Or what does it mean when we have insulin resistance? We have an increase in glucose. We have someone that's diabetic that has hyperglycemia, they have increase in glucose. This means CPT is not working. CPT is not working. We have fatty acid synthesis happening. And then we have decrease in fatty acid oxidation. So we have an increase in free fatty acids. Increase in free fatty acids because we have hyperglycemia, we have increase in free fatty acids. This free fatty acids actually is due to the synthesis of fatty acids. Sorry, yeah, it's happening because we have a lot of glucose. We have a lot of blood. We have a lot of glucose. Sorry, in our body. We're trying to make more fatty acids. We're trying to make a lot of more fatty acids to get rid of it, because we don't want the hyperglycemia. So we have increase in free fatty acids, and we have decrease in the fatty acid oxidation. And that was pretty straightforward. We have increase in this, we're gonna have decrease in this. And then since we have an increase in free fatty acids, and we have an increase in fatty acid synthesis, at that point we would have an increase in the precursor of fatty acids, and that is the malonyl-CoA. Again, recall, what does malonyl-CoA do in this lecture, in this lecture we talked about? It inhibits CPT1. So because it inhibits CPT1, now we have a CPT1 dysfunction, and it's not working since we have a high level of malonyl-CoA. Yeah, so it's the reason, and that is because insulin activates ACC to produce malonyl-CoA. We've mentioned that in fatty acid synthesis, that when we have high number of glucose, we would have high number of insulin, and this high number of insulin would activate ACC, acyl-CoA carboxylase, in order to produce more fatty acids. So acyl-CoA carboxylase actually gets activated by insulin and produces melanoid-CoA. This melanoid-CoA ends up inhibiting CPT1 because we don't need fatty acid. We don't need fatty acid oxidation at this point because we already have hyperglycemia in our blood. Okay. So again, what you guys can already tell that melanoid-CoA inhibiting CPT1 is very, very high yield. So focus on that. Make sure you remember the whole process of why does melanoid-CoA inhibit and fatty acid CPT1, and when it inhibits CPT1, it's inhibiting fatty acid oxidation. Okay, you guys need to understand the whole thing and memorize why it's happening. Second deficiency here is carnitine deficiency. Again, doctor only mentioned it briefly, didn't mention a lot of things that happen. He only said that it's a disorder that happens by age 5. I think age 5 is high yield. If you wanted to bring that question, he's probably going to bring question a baby that is by age like 5, 6, something along those lines. They have cardiomyopathy, skeletal muscle weakness, and hypoglycemia. Hypoglycemia makes sense here because when someone has carnitine deficiency, that means when they are in a prolonged period of starvation or they haven't eaten in a while, they cannot break down those fatty acids to give us ATP or give us glucose. So there is hypoglycemia in our body because there is no carnitine. And again, we need carnitine to break down that fatty acyl-CoA along across those membranes into the mitochondrial matrix. Okay, so here hypoglycemia does make sense, carnitine deficiency due to that. And then they have cardiomyopathy, skeletal muscle weakness. It's not important to go into the pathophysiology of this just yet. It's still early. But yeah, this is just worth mentioning in this lecture as well. Now, CPT2, again, recall that CPT2, what does it do? When we have that carnitine inside the mitochondrial matrix, we have CPT2, which takes the carnitine out and gives us fatty acyl-CoA again, but this time inside the matrix. So, so this is so CP2 deficiency. The most important thing about it, or the most high yield thing to know about it, is it's a myopathy. Myopathy means it's just basically it's a disease that happens in the muscle, so it's related to the muscle. So whenever you see a question that talks about a deficiency in something that has to do with fatty acids, and it produces or it presents with someone having muscle weakness, muscle problems, it's they're most likely talking about CPT2. He mentioned some symptoms. Muscle aches makes sense. It has to do with muscle rhabdomyolysis and myoglobinuria. So it just means blood in the urine right here. And then here they have excessive breakdown of their muscles. Also provoked by prolonged exercise or while fasting or cold or stress. This makes sense again because if they have a deficiency in CPT2, that means they are during times where they're not eating, aka when they are fasting or when they've gone through a prolonged exercise period, they're not eating, so they have low hypoglycemia. When they have low hypoglycemia, what does the body do? The body wants to break down those fatty acids to get ATP and get glucose. But this can't happen when we have a CP2 deficiency, hence why it's provoked by those times. And then it's exacerbated by high fat, low carbohydrate diet. Because when we have a high fat, when we're eating high fat diet, we are ending up storing a lot of fats. This fat cannot be utilized. So again, this is when it gets exacerbated. And I think this is kind of the most high yield thing to know about this disease. Is just CPT2 in your head should always be connected to myopathy. Anything wrong with your muscles, CPT2. Okay. As far as this lecture goes, okay. Now medium chain acyl-CoA dehydrogenase deficiency. So remember, guys, I talked to you about an acyl-CoA dehydrogenase family. So this is one of them. So this is a medium chain acyl-CoA dehydrogenase. So from the name, if it's a deficiency in this enzyme, that means when we get a long chain fatty acid, we are not able to break it down. That means again, we're not going to be able to break down fatty acids for glucose or for ATP. That means we're going to end up with a hypoglycemia period. If the person's not eating, obviously, they're going to end up with a hypoglycemia period, and it's going to make sense that they're going to end up with something called with something called like a they're going to end up with a long chain fatty acids in their blood because or medium chain fatty acids in their blood because this family of enzyme is not working. Okay. Again, same thing. So we cannot hydrolyze those medium chain fatty acids. Patient presents with hypoglycemia, sudden death. So and then this is really important. So this statement here is really important. So since we are not able to break down the fatty acids, we're not able to break down those fatty acids. but then the acetyl-CoA that we are the fatty acids. So as you said, we have fatty acids. They get activated by an acyl-CoA, so they become a fatty acyl-CoA, and then that ends up getting broken down later. So we are still using the acetyl-CoA to bind to the fatty acid to activate it. So this gives us a decreased number of of acetyl-CoA. When we have decreased number of acetyl-CoA, we are not able to produce ketones. So we're going to talk about ketones later, but what's important here to know is acetyl-CoA is the precursor of ketones, and since the acetyl-CoA here is getting used to activate the fatty acids, we have decreased number of acetyl-CoA. This lowers the pyruvate carboxylase. Again, you guys are gonna take this enzyme later in the later lectures, and then this one is gonna limit ketogenesis. So patients would present with low to absent ketones. I guess I can mention ketones now since we're almost there. So ketones is what different parts of our bodies use. So for example, we have the brain, we have muscles, we have different things that utilize ketones. The most important being the brain. So when we're in level, when we don't have a lot of glucose, when we're in hypoglycemic state, we produce ketones, or the liver produces ketones. And then these ketones, they're precursors that acetyl-CoA. So the acetyl-CoA produces ketones. These ketones get utilized by different parts of the body, for example, brain, for glucose or for ATP. So this is a different process than fatty acid synthesis. It's fatty acid oxidation. So it's kind of the same thing, as in like they both try to utilize different things to get ATP, but this one happens in the brain mostly. So you can imagine when we have a hypoglycemic state and the brain cannot produce ketones, it's gonna be a problem, right? And this is why the people present with sudden death, because the brain is not being able to produce ketones to utilize for glucose. And this is important in this deficiency because, as we said, acetyl-CoA is the precursor of ketones, and when we have decreased acetyl-CoA, we have decreasing ketones, and then hence we have the brain's not being able to utilize it. There's two things that is extremely important. As we said, I've already explained why they have hypoglycemia. any question, this is a very high yield disease, not just in mold, but in different in your board exams too. There's two very, very high yield things to understand or to remember is that they have low to absent ketones. So the patient has low ketones, low to absent, and they have C8 to C10 acyl carnitines in the blood. C8, C10, like the number C8 to C10 is very, very important. We've had this in our exam. You guys probably gonna have it in your exam. This just shows you that a medium chain acyl-CoA dehydrogenase breaks down C8 to C10 carbons. Okay, so when we're not breaking them down, we end up having them C8 C10 carnitines in our blood. Okay. Yeah. So this slide where doctor talks about the peroxisomes, it's just kind of like it's out of nowhere. You don't understand what you need to know. So that's why I just kind of summarize it in two sentences. All you need to know is like those two enzymes, whatever, and you move on. Again, so we've already talked about acyl-CoA dehydrogenase. So acyl-CoA dehydrogenase in the peroxisomes does different thing than what it does up there in the mitochondria. So instead of capturing those high energy electrons by ETC as we do in normal mitochondria, what it does, it transfers electrons to O2 to oxygen and gives us hydrogen peroxide. So in peroxisomes, the acyl-CoA dehydrogenase transfers electrons to oxygen and gives us hydrogen peroxide. Okay. And in the peroxisomes, we have an enzyme called catalase. This enzyme can actually catalyze that hydrogen peroxide that this enzyme just produced. So it catalyzes this enzyme into water and molecular oxygen. Okay, so hydrogen peroxide, as you guys know, is pretty dangerous. So it's just telling us that in the peroxisomes, acyl-CoA dehydrogenase actually gives us hydrogen peroxide. But good thing, in the peroxisomes, we have something called catalase, because this catalase can actually take that hydrogen peroxide and break it down into something that is that is used in the body or that can get utilized in the body, and that is water and oxygen. Okay, that is all we need to know from that slide. Okay, so let's say I've mentioned already that when we talk about fatty acids, we get something like 14 carbon fatty acids, we get something like 16, we get something like 12. Okay, what if we get something like 13? We get an odd number kind of fatty acid. So goes through those four different reactions. From 13 it becomes 11, 11 becomes 9, whatever, until it becomes 3. Or 3 or 1, whatever. Sorry, yeah, like it keeps coming down until it becomes 3. So when it becomes 3, what are we supposed to do at that point? It cannot be broken down into it's because it's an odd carbon, it's an odd chain fatty acid. It needs additional enzymes that we haven't talked about earlier. So when a propionyl CoA is a 3-carbon fatty acid, and this propionyl CoA gets catabolized to succinyl CoA, and that succinyl CoA can go in the TCA cycle. Okay, so when you have something like an odd chain fatty acid, we call it a propionyl CoA, and then when we have that propionyl CoA, what we try to do is make it a succinyl CoA, just so it can go inside the TCA cycle, and then through the TCA cycle, it can give us what we need, which is ATP, and then the different kind of products of TCA. So in order for it to become a succinyl CoA, it goes through 2 different reactions. Those 2 different reactions are very, very important. Again, not just in this lecture, but for your future exams, your board. We get 2 enzymes. We get a propionyl CoA carboxylase, and we get a methyl malonyl CoA mutase. Okay, so here you can see we get, when we have an even number of fatty acids, whatever, we are good to go and do our different reactions. When we have an odd carbon fatty acid, that is when it goes through that's when it goes through propylene CoA. That's when it comes to propylene CoA. It goes through those different enzymes, those two enzymes, and then gives us succinyl CoA that goes in the TCA cycle and gives us the results we need. So the first enzyme that goes through is the propionyl CoA carboxylase. That enzyme requires biotin or B7. So B7 is the same thing. It's important to know that biotin is B7 because sometimes they try to trick you by bringing, like, instead of bringing biotin, they bring like B1, B3, B6, B7, which one's correct? Yeah, so you need to know which enzyme or coenzyme stands for which number. So B7 is biotin. So first of all, it goes through this enzyme requiring biotin, and then it goes through methylmalonyl CoA mutase. This enzyme requires vitamin B12 or cobalamin. This is the same two names for the same thing. So propionyl CoA carboxylase requires B7 or biotin. Then we get methylmalonyl CoA. From methylmalonyl CoA, we have the enzyme called methylmalonyl CoA mutase or B12, which is cobalamin. This gives us succinyl CoA. Succinyl CoA can get utilized later and give us oxygen from the citric acid cycle. Okay, so this happens when we have an odd chain fatty acid. Okay. We are down to ketogenesis. So far we've talked about fatty acid oxidation. We mentioned everything that goes. Since we get a triglyceride all the way down to a long chain fatty acid, and then down to the 4 different reactions to give us the ATP we need. So here we're going to talk about ketogenesis. So I've already kind of briefly went over it up there. But here we I've already went over it up there kind of briefly, but here we're actually going to talk about it. So there's ketogenesis, ketolysis. So the making of the ketogenesis, the making of ketones or ketogenesis happens in the liver. So this is a very important thing to know, is that ketogenesis happens in the liver. Okay. We have a fatty acyl CoA. As we said up there, at that point we have a fatty acyl CoA, right? And that fatty acyl CoA is, if some of it goes through and does those 4 different reactions. So fatty acyl CoA goes through those different reactions to give us the acetyl CoA, fatty acyl CoA. And then keeps getting broken down until we get ATP, or it can go through ketogenesis, and that is when fatty acyl CoA gives us something called acetone and 3-hydroxybutyrate. So these can get, again, utilized in the brain and different extrahepatic tissue for energy. So the making of ketogenesis, the making of ketones or ketogenesis. What's important to know here is the fact that when does that happen, or when ketones, when do we start producing ketones? Is when we have a low oxaloacetate state. So when there's low oxaloacetate, this is when the acetyl CoA would not be able to enter the TCA cycle, or the citric acid cycle is the same thing. And then because of that, it actually goes and forms ketones instead. So we have broken down the fatty acids, and we are ready for the fatty acids to enter the citric acid cycle and give us energy. If we already have done all of that, and then we actually end up getting a low oxaloacetate state, then at that point we cannot go through the citric acid cycle. That is when the fatty that is when the that fatty acid CoA goes and produces ketones instead. Okay, so in a low oxaloacetate state is when ketones are made. That kind of, yeah. Um, so what's important to know here? Keep Ignore all those different enzymes. There's one enzyme that really matters, and that is the HMG CoA synthetase or synthase. This is the rate limiting step. This is the rate limiting step of making ketones in the liver. So what we need to understand here is, or what we need to memorize is this enzyme right here. The fact that ketogenesis happens in the liver, and the fact that it happens when there is low oxaloacetate. Okay. Okay, now we have, again, we broke down fatty acids. There is low oxaloacetate, so we can't go into the citric acid cycle. We went to the liver, that fatty acid CoA went to the liver to produce ketones and go through ketogenesis. Now we have ketones. What do we do with it? So again, in the periods of fasting, when there is no oxaloacetate enter the citric acid cycle, we make ketones. Now we need to utilize. We cannot produce energy. We can't break down. We can't break down the energy. We can't utilize our fatty acids for energy because there's no oxaloacetate. We need to utilize the ketones now since we need the energy. So where does that happen? It happens in extrahepatic tissue, especially the brain. That is when we utilize the ketones to give us acetyl-CoA. Then acetyl-CoA can give us the energy we need. So it only happens, or not only, it happens in the extrahepatic tissue, and that's for a reason we're going to talk about in a second. It happens in the brain and different peripheral tissue, and that's muscle, for example. We get breakdown of ketones. So we have already gone through this whole thing that's happening in the liver. We get something called hydroxybutyrate. Oh, and there's something to mention here also. So acetone. You can already tell that this is the smell of alcohol. Or we call it acetone usually. This is why sometimes when people are fasting, they get like a kind of acidic breath, and that is because at that point we've been in a prolonged kind of fasting period. We are utilizing these ketones, or we are producing these ketones, and that's why we have that acetone smell or that like acetone breath in our mouth. So we have already gone to the fact that we have hydroxybutyrate from the synthesis of ketones in the liver. Now we need to utilize it. So again, a lot of those enzymes we don't really need to understand or memorize. The most important enzyme we need to memorize is thiophorease. So thiophorease is an enzyme that is used in the breakdown of ketones or ketolysis to give us acetyl-CoA. So this enzyme is not found in the liver. So because it's not found in the liver, this means that the liver cannot utilize ketones. So it only produces ketones, but then it only gets utilized in tissues that have the enzyme thiophorease. That is the liver, the brain. There's different tissues that we're not going to talk about. Not really worth mentioning, but ketones cannot be utilized unless the tissue has thiophorease. And that is why in the muscle, when it's a prolonged kind of fasting period, we can utilize the ketones through thiophorease to give us acetyl-CoA that can then enter the citric acid cycle and give us the energy we need. Okay. That is all I have for today. That is beta oxidation and ketone synthesis. Inshallah, it was helpful. Again, I've kind of went over the high yield stuff. I tried to include everything in my lecture that the doctor talked about and I saw is important to know for your exam. Um, Again, I really wish it wasn't recorded. I really wish I could have done it live. Then I would have been able to ask you guys questions and then have it interactive. Other than that, it was an easy lecture. Alhamdulillah. I hope it was helpful and please go over Lippincott and integrative biochemistry textbooks to solve those questions after this lecture. Um, and I've left my contact information if you guys have any questions. And yeah, thank you so much for watching and let me know if you guys have any questions. Thank you.